Hello and welcome to our PKSim online tutorial series. Please be reminded that the results of the checkmark videos are necessary to follow the content of this video properly. In this video we will show you how to create an individual in PKSim. In detail you will see how to define body dimensions, change the anatomy and physiology, add metabolizing enzymes or transporters, as well as how to clone and scale your individuals. To create an individual, right-click on Individuals in the Building Block Explorer and choose Add Individual. Enter a name for your individual. In the tab Biometrics, you can define the body dimensions of your individual. A human male European is predefined with mean values for age, weight and height, informed by the ICRP database available within PKSim. In the section Population Properties, you can choose another species, population, or gender. In the section Individual Parameters, you can define age, weight and height depending on your data. If you don't know the full characteristics of your study individual, you can go with the database average values for the missing information. By clicking on Mean, the values reset to the predefined mean individual. Then click Next. The second tab is called Anatomy and Physiology, where you can see database values regarding individual anatomy and physiology. Make sure your parameter layout is set to Advanced. Here you can change, for example, individual GFR values or settings for enterohepatic circulation, depending on your research question. Search for GFR in Anatomy and Physiology. Then click Next. The third tab is called Expression, where you can add metabolizing enzymes, transport proteins and protein binding partners. To add a metabolizing enzyme, right-click on Metabolizing Enzymes. There, choose Add Metabolizing Enzyme Database Query to connect to the PKSim Gene Expression database. A window Add Enzyme Expression is opened. The first tab is called Protein Selection. Here you can enter the name of the enzyme you would like to add to your individual, for example CYP3A4, and click on Search. Database entries matching your search request are shown. By clicking on Gene ID, the corresponding database entry is opened for you to check if you have found the right enzyme, transporter or binding partner. Double-click on the enzyme you would like to add. The tab Expression Data Analysis shows expression profiles for your chosen protein, determined with different methods. Click Next. In this tab, you can choose the expression profile you would like to use in your individual with the radio buttons in the upper part. After choosing RT-PCR, click OK. Now you can edit the enzyme entry and change predefined values if necessary. In the section Properties, you can edit the reference concentration, which corresponds to the concentration of your protein in the organ with the highest expression, as well as the protein half-life in liver and intestine. For many CYP and UGT enzymes, you can find ontogeny information in the PKSim ontogeny database. If no ontogeny is predefined for your enzyme, choose Undefined. In the section Localization, you can choose where your enzyme is located in the cells and in vascular endothelium. In the lower part, you can see the enzyme expression in different organs relative to the organ of highest expression. You can change the relative expression by entering new values. When you are finished editing your individual, click OK. The individual appears on the left-hand side. If you right-click on this individual, you have different options. You can edit your individual. Biometrics cannot be edited. Changes in anatomy and physiology and expression can be performed as described before. You can rename your individual. Click Rename, enter a new name and click OK. You can clone your individual to create an individual with the same properties. Click Clone, enter a name and click OK. With the function Scale, you can also reproduce your individual, but editing the biometrics is possible using this option. Click on Scale and the window Scale Individual will open. You need to name your new individual. Here the first step again is biometrics. The properties can be changed as described previously. Click Next. The second tab is called Scaling Configuration. Previously edited parameters, such as specific GFR, are listed. Here you can decide if you want to revert to the original database value by choosing Use Default Value, or the edited value of the individual you scaled from, choosing Use Source Value for the new individual. Click Next. In the next two tabs, you can edit the anatomy and physiology and the expression parameters as previously described. The last option is the deletion of your individual by clicking on Delete and confirming the deletion. Many thanks for watching.